culture, we have a special session on light reaction of photosynthesis. Here we have Chef Clover giving a lesson to a young plant, Blossom.
We're doing a very special dish that has been passed down from generation to generation. But before we start, let me introduce my volunteer. Her name is Blossom. Hey Blossom, how are you?
Not so great, Chef. I'm so famished and I think my ATP are on E.
Aww.
Well, good thing you're here. Today we're doing a segment called Cooking with Light Ingredients. It's how we make ATP and NADPH. Did you know you can make your own ATP?
Really? I had no idea. How great is that in this economy?
It's great. First, we need to add a dash of sunlight to activate the chlorophyll molecules on your receptors of your chloroplast.
Awesome! I'm feeling better already.
The sunlight will then excite your electrons by absorbing mostly blue and red light. This begins the process known as photooxidation. This creates the energy that needs to be passed to the photosystem to make. In your chloroplast, you have two complexes, photosystem 1 and photosystem 2. Photosystem 2 funnels all that tasty energy created to pheophylline then to plastoquinone, which makes the reduction of plastoquinone. After you have all that yummy reduction of plastoquinone, you need to make up for the 2 electrons that were lost. So you need to add a little water just to replenish them. From this, you transfer the 4 protons to the lumen.
Wow, that's easy. I can do that.
When you add the water, photosystem 2 will split it and oxygen will be released. We will set photosystem 2 aside for a while and let it simmer a little bit. Now we can go to the next station, which is photosystem 1. But in order to get there, we need to strain the electrons through the strainer. The strainer is called your cytochrome B6F complex and is located between the 2 photosystems. Straining the reduction of plastoquinone causes the plastoquinone to be oxidized 1 electron at a time. The results are a reduction of plastocyanin and the release of 4 protons.
Wow, that's a lot to process, but I can't complain since I don't have much work to do.
Okay, now that you have the reduction of the plastocyanin, you transfer 2 electrons to photosystem 1 and you are on your way.
This is really starting to smell delicious.
I couldn't agree with you more. Now in photosystem 1, you have a P700 group that can accept electrons from plastocyanin. In P700, here you add another dash of sunlight to excite it again, and it flows spontaneously to other chlorophyll molecules, such as quinones and iron sulfur clusters. After you accept the electrons to P700, you transfer those electrons to ferredoxin, where they will be reduced again, 1 electron at a time to the iron sulfur clusters.
I think I kind of get it now.
Great. Okay, now that you have reduced the ferredoxin, you go into 2 separate pathways, cyclic and non-cyclic. I will explain the cyclic pathway first. You have an enzyme called ferredoxin NADPH reductase, and it will reduce NADP plus to NADPH. For every molecule of water that you have present, you get 2 NADPH. Let's move to the cyclic flow now. In cyclic pathway, the electrons are not used to reduce NADP+, but instead they are to return to the cytochrome B6F complex.
Can you explain that?
Sure. The electrons that are transferred to the plastocyanin will flow to photosystem 1 to reduce P700.
But why can't I just use the electrons to make NADPH all the time?
Well, that's a very good question. See, by alternating between cyclic and non-cyclic pathway, your photosynthetic cells can make either ATP or NADPH, according to what they need.
Well, that makes sense, because if I have too much of something, I can store it to use later instead of wasting energy to make more.
Right. By returning the electrons to the cytochrome B6F complex, you build up a proton gradient so you can finally get those ATP much desired.
Great. Can we get started now?
Of course we can. To make ATP, you need the CF1 and CF0 complex, which has two components. The CF1 components is where the ATP synthesis occurs. Do you remember the proton gradient that was built before?
I was wondering where those went.
Well, wonder no more. I'm glad you asked that. In non-cyclic flow, 12 protons were generated to produce 3 ATP. That's all you need to produce the other molecules.
So there's other things I can do with my ATP?
Yes, but you have to stay rooted for next time. We are doing a session on the dark reactions of photosynthesis, where you can use those ATP and NADPH you use to make tons of other goodies.